Hello everyone. So today I'm talking about a very interesting bacteria, which is Bacillus anthracis. We all know about the disease anthrax, isn't it? Anthrax is an ancient disease, and many researchers believe we can trace it all the way back to the 700 BC in ancient Egypt. And this Bacillus anthracis has many medical importance. It is the first bacteria isolated in pure culture and shown to have spores. And many greatest microbiologists like Robert Koch studied about this bacteria. And Robert Koch developed his famous Koch postulates using this bacteria. Okay, and it is the first attenuated vaccine, and it was discovered by the greatest microbiologist Louis Pasteur. So this is a very important and interesting bacteria, of course. So now we know anthrax is not contagious, okay? But what does it mean? It means it doesn't spread from person to person, okay? So how can it spread actually? Actually, anthrax is a disease of livestock, and it ingests, and that livestock actually ingests spores in plants, soils, etc. And how it can enters into the human when humans handle infected animal products like wool, leather, etc. They can be infected when humans inhale anthrax spores from infected animal products or eat undercooked meat of infected animals. They can also be infected with Bacillus anthracis. So this is actually a cycle. This cycle continues, and Bacillus anthracis Bacillus anthracis produces the disease anthrax. Okay, now we're talking about some important transmission features. Like we know it is a spore forming bacteria, but the spores are very highly resistant, and they resisted the dry heat almost 140 degrees Celsius, even for 1 to 3 hours. Okay, boiling for 10 minutes, they can resist this high temperature. And if we want to kill it, we have to use autoclaving. Okay, so this is a highly resistant spore forming bacteria, and the soil is contaminated with their spores from generally carcasses of dead animals, and the spores remain viable for decades. So, contact with infected animals or with their hides, hair, bristles, through trauma to skin is the source of infection in humans. Okay, and another, the spores can also be inhaled into the respiratory tract. The pulmonary in anthrax then occurs. And also, there is gastrointestinal anthrax that occurs when contaminated meat is ingested. Okay, now we'll talk about very important here, virulence factors. Sorry. There's another slide. Is this percentage of occurrence? Cutaneous anthrax is very common. Almost 95% cases, and pulmonary anthrax it is only 5%, and gastrointestinal anthrax is very rare in humans. Okay, now the virulence factors. So there are two main virulence factors. They are capsule and exotoxin. And the capsule inhibits opsonization and phagocytosis. And antibodies to capsule are not protective actually. And the exotoxin, it is mainly composed of three main components: the edema factor, lethal factor, and protective antigen. When edema factor combines with protective antigen, it is called edema toxin, and when lethal factor combines with protective antigen, it is called lethal toxin. So, what are their functions? The edema factor actually is an adenylate cyclase, and it causes an increase in the intercellular concentration of CAMP. So, what is CAMP? Cyclic adenosine monophosphate, CAMP. And this causes an outpouring of fluid from the cell into the extracellular space, which manifests as edema. That's why it's called edema factor. Okay, edema factor. So, and now lethal factor. Little factor actually is a protease and it cleaves the phosphokinase. So what does the phosphokinase do? It activates the MAPK signal transduction pathway. So what is MAPK? It's mitogen activate protein kinase. Mitogen activated protein kinase. And this pathway controls the growth of human cells. And cleavage of this phosphokinase inhibits the cell growth. Okay, and last of all, the protective antigen. Actually, it forms pores into the human cell membrane that allows edema factor and lethal factor to enter into the cell. That's why it's named protective antigen. No, protective antigen actually refers to the fact that antibody against this protein protects against the disease. That's why it's called protective antigen. Okay, so now the very important part, pathogenesis. We know there are three kinds of anthrax: cutaneous, pulmonary, and gastrointestinal. So, coming to cutaneous anthrax pathogenesis, the spores germinate in the tissues at the site of entry, and the growth of vegetative organisms results in formation of gelatinous edema and congestion. And then starts the toxin production. And the toxin, this is called toxemia. Here is a short briefing of the pathway of toxin, edema toxin and lethal toxin. Edema, ATP produces, and then CAMP, and then reactive oxygen intermediates. Okay, and another lethal toxin, it controls MAPK pathway, and it controls the growth of human cells. Okay, so then shock, death, and associated features actually occurs. So actually, it's a very it's like a difficult pathogenesis, but if we want to short it out and if we want to remember it very easily, then we just have to remember this it's like two main important toxins, edema toxin, lethal toxin, and the basic associated pathogenesis. Okay, now coming to our intestinal intestinal actually it's very rare, so we don't need to remember this gastrointestinal pathway. Just I'm talking now pulmonary anthrax pathogenesis. So it's an inhalation anthrax, right? And it's called wool sorter's disease because the spores from the dust of wool, hair, or hides are inhaled. That's why it's called wool sorter's disease. Okay, then it is phagocytosed in the lungs and transported via lymphatic drainage to the mediastinal lymph nodes, where the germination occurs. And this is then this is followed by toxin production and the development of hemorrhagic mediastinitis, sepsis, which is very rapidly fatal actually. And another septicemia, toxemia, and shock, death, this kind of associated features will occur gradually. Okay, now the clinical findings. The clinical findings actually are cutaneous anthrax. So the typical lesion of cutaneous anthrax is painless ulcer. Here we are seeing a ulcer, and this ulcer has what do you think? It's a black color, so it's called black ishker. Ishker means crust, scab. Okay, and this lesion is called malignant pustule, and if it is left untreated, it progresses to bacteremia and also death. So this is black ishker, which is happened in cutaneous anthrax. And this lesion starts as a papule 1 to 7 days after infection. The lesion is then surrounded by an area of edema and erythema due to multiplication of the organism. And the lesion rapidly evolves into a vesicle, and the fluid within the vesicle turns black due to hemorrhage into that area. And the lesion finally ulcerates and develops a black ashker after 7 to 10 days, okay, with surrounding edema. Okay, and note that this necrotic lesion covered by crust is caused by lethal factor, okay, and the surrounding area, which is edematous area, this is caused by edema factor. Edema factor caused edema, okay, edematous surroundings. Okay, Now, we'll just talk about the clinical findings of pulmonary anthrax and gastrointestinal anthrax, but they're less important actually. So, pulmonary anthrax, the incubation period is 1 to 3 days and maybe as long as 6 weeks, followed by sudden onset of symptoms. Actually, it resembles influenza, especially a dry cough. Okay, and this rapidly progresses to hemorrhagic mediastinitis, bloody pleural effusions, septic shock, and death. Okay, and although the lungs are infected, the classic features and extra picture of pneumonia are not present. Mediastinal widening is seen on chest X-ray. So it's an important diagnostic criteria actually. Mediastinal widening on chest X-ray. So and hemorrhagic mediastinitis actually severe life-threatening complications actually. So this you have to memorize about pulmonary anthrax clinical findings. And now gastrointestinal anthrax, the symptoms are vomiting, abdominal pain, bloody diarrhea, this. And the incubation period is nearly 2 to 7 days. But the fatality rate is very high. The death generally occurs within 2 to 5 days after the onset of fatal cases. Okay, fatal cases. Okay. Now, laboratory diagnosis. Actually, the specimen it includes fluid aspirated from cutaneous lesions, and sputum, cerebrospinal fluid, and blood for culture. Okay. So here's the picture of Bacillus anthracis. So here we can see the morphology of Bacillus anthracis. It's a large gram-positive, non-motile bacillus, and it's square ends. It has square ends and frequently found in chains. So the square ends are found in chains, and it's a very actually unique property. What is it? The unique property is that we know the capsules of normal bacteria are polysaccharides. Okay, but the capsule, antiphagocytic capsule of Bacillus anthracis is composed of D-glutamate. What I'm saying? D-glutamate. So this is the unique property of Bacillus anthracis. The antiphagocytic capsule is composed of D-glutamate. Okay, and another very actually interesting property. Okay, here's a picture. It is called Medusa. It's a character of Greek myths, and just to see how her hair is. So this is the picture of Medusa, and this is a picture of blood agar culture of our the culture of Bacillus anthracis in blood agar medium. So can you find any kind of similarities between these two? Okay, maybe you cannot find, but actually, the age of this colony resembles locks of matted hair. This colony resembles locks of matted hair, and that's why it's called Medusa head. Okay, but actually, it's weird, isn't it? Because the any head with curly hair will look like interlacing bacterial chains. Okay, so why is it Medusa head? Actually, many people like to glorify medicine with fancy names. That's why it's called Medusa head. Okay. Okay, so it's a very important characteristic. It is the Medusa head in blood agar culture. Nutrient broth, gelatin stab culture, they can also done, and biochemical reaction are also done, but it is of very little value. And now definitive identification is also done by PCR or ELISA test. Okay, now the treatment. The treatment is ciprofloxacin. Ciprofloxacin is our drug of choice, but doxycycline is also it's an alternative drug. It can be used. Okay, so here actually we're coming to the end of our lecture. So now here is a quiz for you. So let's start. A 37-year-old male is admitted to the hospital due to 3 days of fever, shortness of breath, and recent episode of hemoptysis. He works as a shepherd during the summers, has no smoking history, and denies any recent travel. A sputum gram stain reveals gram-positive rods, which of the following is most likely true regarding the microorganism? So option A, the polysaccharide capsule is antiphagocytic. One component of the exotoxin complex results in increased levels of CAMP. The toxin inactivate elongation factor EF2 and it is an obligate anaerobe. Okay, so where's the answer? So, or if we read thoroughly this question, so we can all, of course, we can find that this indicates the microorganism Bacillus anthracis. Okay, so Bacillus anthracis, we know it's a aerobe. Okay, so Bacillus anthracis, so D false, C false, and A, of course, is false. It's made of diglutamate. So last one is left option B. So B, we know the edema factor is increases the levels of CAMP. It's an important feature. So the answer of this quiz is B. One component of the exotoxin complex results in increased levels of CMP. Actually, if you can figure out this microorganism is Bacillus anthracis, then of course you can answer the quiz. Okay, so you first have to figure out what microorganism it is indicating. Okay, so that's the end of our lecture. So thank you all. Bye bye.